Hi, I'll be teaching today about the steps in genetic engineering and cloning. The photo you see in the lower left is of Dolly the sheep. She was cloned in 1996. And this chapter is on genetic engineering, which is basically altering the genes of organisms for practical purposes. Genetic engineering can be beneficial, but at the same time it can be controversial as well. Some of the main organisms used in genetic engineering are bacteria. The reason being they are fairly simple and they have a circular ring of DNA called a plasmid in which scientists have found out how to alter it. They use what's called restriction enzymes which cut DNA kind of like a scissors at certain locations. The main one we will be speaking about is EcoR1, the lower one there, which cuts when you have the bases AAT. And they use genetic engineered bacteria to make many useful pharmaceutical products such as insulin, for diabetes of course, growth hormone, erythropoietin, Taxol, which is an anti-cancer medicine, and other drugs too. The steps in genetic engineering bacteria are pretty straightforward. They first involve dissolving the cell membrane and cell wall of the bacteria. And then they cut the plasmid again, which is a ring of DNA, with restriction enzymes. Again, this one is EcoR1, which cuts between AATT. And once they cut the plasmid, it opens up. It's a ring, but it opens up. Then they cut a human gene in this case. We'll say it might be the insulin gene with the same restriction enzyme. So it also has the exposed spaces AATT. They then mix the two together, and in the picture the human insulin gene is in pink and the plasmid is in gray. They also have to add a chemical called ligase, which makes the two pieces stick together, and then you form what is called recombinant DNA, which is, the name implies, DNA combined from two different organisms. This is another diagram showing the steps in genetic engineering. Again, for our example, we're going to say this plasmid is used to make human insulin. And so if you look on the left, there's a human cell, and they cut the insulin gene out with a restriction enzyme. On the right top is the bacterium, and they cut the plasmid with the same restriction enzyme. And then they basically add ligase once again, and that forms this recombinant DNA, which has a human insulin gene inserted into the plasmid ring. They then let the bacteria reproduce, but not all the bacteria will have this recombinant DNA in them, so then they have to identify which ones have the recombinant DNA and which ones don't. Again, they let these bacteria reproduce, and as their populations grow, they will then make insulin, which is cheaper than insulin was formerly. It's also better quality. This slide is showing you the steps in cloning. In this case, we're going to show how Dolly the sheep was cloned. They first have to get an egg from a sheep, and then they take the nucleus out. That is called enucleation, or an enucleated egg. So it does not have any genetic material in it. They then take the sheep they want to have cloned and get any cell from it except for an egg or sperm. It has to be a somatic cell. In this case, it's the mammary cell. They then put the mammary cell with a nucleus in it, which remember is diploid, and put that adjacent to the enucleated egg. They apply electricity, which I believe creates little tiny holes in the cell membrane, and so that nucleus then can enter the cell. They also sometimes use a very, very small needle to inject the nucleus into the enucleated egg. They then let it develop, I believe, a few days in a petri dish, and then they have to put it in a surrogate mother, in this case any female sheep, in which the egg develops. And after 5 or 6 months, which is a gestation period for a sheep, A lamb will be born that is a clone of the sheep in which they got the somatic cell from. So those are the steps in cloning. Thanks for listening. Bye.